You know, the entire vast world of bacteria, everything from the good guys in your gut to the really nasty bugs, can basically be split right down the middle into two massive groups. This simple division is one of the most important concepts in all of biology and medicine, and it all starts with a very simple question.
And it all comes down to this. When scientists use a specific staining technique, some bacteria turn this deep, rich purple, while others end up looking bright pink or red. So what is going on here? What does this simple color difference actually tell us about these microscopic life forms?
To answer that, we actually have to take a little trip back in time, all the way to 1882. A Danish bacteriologist named Hans Christian Graham came up with a method that would bring this fundamental difference to light. And believe it or not, we still use it every single day in labs all over the world. This technique, of course, is called Graham staining, and its whole purpose, its entire reason for being, is to tell bacteria apart based on one key feature, the structure of their cell wall. You can almost think of it as a test of their microscopic armor. The process itself is basically a straightforward four-step recipe. First, you dose all the bacteria on a slide with a purple dye called crystal violet. Then, you add iodine, which acts like a fixative. It basically grabs onto the purple dye and forms this big complex, locking it in. Then comes the most important step, the decolorizer. And finally, you add a pink counterstain, like saffronin, to finish it off. Okay, this right here, this is the moment of truth. That wash it with alcohol or acetone, it's the great divider. Some bacteria just stubbornly hold on to that purple dye no matter what, but others, it washes right out, leaving them clear and ready for that pink stain. And that leads to the crucial question, why? And here it is. This is the big reveal. The reason for that color difference isn't some random chemical quirk. Nope. It's a fundamental and architectural difference in how they're built, specifically the design of their cell wall. And this illustrates the difference perfectly. So think of the gram-positive cell wall like a really thick, multi-layered sponge. It's made of this stuff called peptidoglycan, and it just soaks up that purple dye complex and holds on tight. The gram-negative cell wall, on the other hand, is a totally different story. It's like a very thin sponge that's protected by this slippery, lipid-based outer membrane, almost like a raincoat. Let's dig into the details here. This table breaks it all down. I mean, just look at the cell wall thickness. Gram-positives are rocking a massive 20 to 80 nanometer thick wall. Meanwhile, gram-negatives have this tiny 8 to 10 nanometer one. But the real game changer, the thing you have to notice, is that outer membrane. It's only on the gram-negatives, and it contains this unique substance called lipopolysaccharide, or LPS. So here's how that plays out. For the gram-positive bacteria, when you hit them with that alcohol wash, it actually dehydrates that super thick peptidoglycan wall. It kind of shrinks the pores in the wall, and that physically traps the big purple dye complex inside. That purple color is locked in tight. It's not going anywhere. But for the gram-negative bacteria, the alcohol decolorizer does something completely different. It just dissolves their outer lipid membrane, that raincoat we were talking about. And once that's gone, it exposes the super thin peptidoglycan layer underneath, which just can't hold on to that big purple dye complex. It washes right out, leaving the bacterium totally colorless and ready to soak up that pink saffronin stain. Okay, so we get the how. Purple, pink, cell walls, got it. But why does this matter so much? I mean, who cares, right? Well, this is way more than just a neat lab trick. This structural difference has huge, profound consequences, especially in medicine. This is literally about knowing your enemy. And here it is. This is the absolute crucial point. That outer membrane on gram-negative bacteria, it's not just a shield against stains, it's heavy-duty armor against our antibiotics. It makes the cell wall incredibly difficult for many drugs to penetrate. This is why, as a general rule, gram-negative bacteria are naturally more resistant to antibiotics and can be way, way harder to treat. Let's put some familiar faces to these names. When we're talking about gram-positive bacteria, you're thinking of things like Staphylococcus. Yep, the bug that causes staph infections. And Streptococcus, the one responsible for strep throat. These are definitely serious pathogens, but that cell wall structure of theirs gives our drugs a way in. And now for the gram-negatives. This group includes some pretty notorious characters, like E. coli and Salmonella from food poisoning, but also species that cause really serious conditions like meningitis, pneumonia, and urinary tract infections. That extra layer of defense makes them incredibly formidable opponents. And the differences don't stop there. It's also about how they attack.
Gram-positive bacteria typically produce and secrete toxins, called exotoxins, out into your body. But gram-negative bacteria, their toxins, called endotoxins, are actually part of their physical structure, baked right into that outer membrane. This means when the bacterial cell dies and breaks apart, it can release a flood of these toxins, causing severe symptoms like fever and shock.
So, when you see all these critical differences in antibiotic resistance, in toxin production, it becomes really clear why telling these two groups apart and telling them apart fast is so vital. In a hospital, the gram stain isn't just an interesting experiment. It's a frontline diagnostic tool that helps doctors make decisions that can literally save lives.
And it's amazing how much information a clinician can gather in just a few minutes. They're not just looking at the color, but also the shape and how the bacteria are arranged. So are there little purple spheres clumped together like grapes? That immediately suggests staph. Purple spheres in a nice little chain? Probably strep. Pink rods? Well, that could be E. coli. This gives them a massive head start on choosing the right antibiotic, days before other lab results come back.
Yeah, and this quote from Stat Pearls just hits the nail on the head. In the fight against any infection, speed is everything. The gram stain delivers that first, critical piece of intelligence to guide treatment right from the very start. So just think about that. A simple staining technique from the 1880s reveals this fundamental divide in the bacterial kingdom that still dictates how we fight diseases today. It's an incredible testament to brilliant science, but it also leaves us with a really big question for the future. As these bacteria continue to evolve and keep developing new ways to resist our drugs, how are our century-old methods going to have to adapt to keep up?